Hello everyone, I'm Sheena and welcome to my channel Comprehend Biotech. We are discussing RNA sequencing and this video is part 2 of the session on RNA-seq. In part 1, we have already discussed RNA isolation and species selection. We have started into the second step, that is the library preparation step. In today's class, we'll be learning more about adapter ligation and also about Bridge PCR. So let's start. Welcome again. As mentioned, part 2 of RNA sequencing starts now. So in part 2 today, we will be discussing the basic PCR reaction, adapter ligation, why adapters, basic elements in adapters, and Bridge PCR. So all these topics will be covered today in this session. Okay, so we are talking about library preparation. We have fragmented our RNA. We have reverse transcribed it into cDNA. Now, before we go into the next step of adapter ligation, you're going to add adapters, right? I just wanted to talk a bit about the technique of PCR, okay, polymerase chain reaction. In that, basically the 3 steps, that is denaturation, annealing, and extension, just so that, using this concept, we can just go forward so that I don't have to discuss it later, okay? So the concept is only this much. You have your double-stranded DNA. First of all, you will denature it, right? Denaturing will make these double-stranded become single strands. Next, you will add primers to it. These primers will be going and attaching themselves to the 3' end of the strands. So over here, this primer, which is complementary to the ends, right? It will come and add over here. This primer to this 3' end of the second strand, okay? And in the third step, extension happens in the presence of DNA polymerase. Specifically in PCR, we use stack polymerase, which is a heat tolerant DNA polymerase, okay? So this will be extended, and that's how you actually, from one double-stranded DNA, you get 2 double-stranded DNAs, or 2 copies you get. That is what happens in polymerase chain reaction. This is repeated again and again, numerous times. Many times these cycles are repeated. And from 2, you will have 4. That's how it goes, okay? So I just wanted you to be clear on this. Why on 3' in the primer is coming and adding? Because always you will notice that the new strand of DNA, whenever it is synthesized, it is synthesized in the direction 5' to 3'. So you can see over here, if this is a new strand that is going to be synthesized, it will be from 5' to 3'. Same way over here, the new strand is always synthesized from 5' to 3'. So your primers are always going to come and attach on the 3' end of the old strand. Okay, I know these are very simple things, but sometimes we tend to forget them. That's why I'm just reminding it over here. Okay, so now we come to the important part of library preparation, which is adapter ligation. Okay, with the name itself, you understand you're going to ligate or attach adapters on both ends of your cDNA. Okay, so now you have your cDNAs. The cDNAs, before you're going to add the adapters on both ends, right? You need to do something called end polishing, okay? Because sometimes when the fragmentation of mRNA occurs, or during the cDNA synthesis itself, sometimes you might have overhangs left over here on the 5' end or 3' end, okay, of the cDNA that you get. So we do not want overhangs or sticky ends, okay? We want blunt-ended molecules, okay, DNA molecules, cDNA molecules. So in order to do that, you need to carry out end polishing. And the enzymes that help in that are T4 DNA polymerase, and clinofragment, and T4 polynucleotide kinase. Basically, what they do is they fill the 5' overhangs by polymerase activity. So if there's a overhang of 5', its complementary 3' strand will be filled by T4 DNA polymerase. And if there is a 3' sticky end, which is kind of overhang which is coming up, that will be cut off by this clinofragment, basically. Okay, and that is done by the 3' to 5' exonuclease activity. And T4 polynucleotide kinase, what it does is it adds phosphates to the 5' ends. Why that is necessary? Because if some adapter has to come and attach to your DNA fragment, there has to be a 5' phosphate available. Only then the bond will be formed. How is a bond between 2 nucleotides formed? You have a 3' OH and a 5' phosphate, right? From one nucleotide, you have 3' OH. From another DNTP, you have, or nucleotide, you have the 5' phosphate. 3' OH, 5' phosphate forms a phosphodiester bond. And that's how 2 nucleotides join together in a strand of DNA. Okay. Now, Adapter ligation. First of all, why do we need to ligate adapters? Basically, DNA sequences are unknown. Unknown means we do not know the sequence of the DNA, right? So two different adapters need to be attached. Just to clear this point, let me go back to that diagram that I was explaining you about PCR. If you look over here, during PCR, we need to have information about what the ends of the DNA fragment look like. And generally, we do have that information. And that is how these primers are designed. Or we generally attach something, okay, and complementary to that, your primers are designed. In this case, since the whole topic is RNA sequencing, that means we still do not know the order of nucleotides. We do not know how the RNA looks like. We have converted into cDNA. We do not know what nucleotides are there. So how do we design primers for it, right? We cannot design primers on our own by it. So what we need to do is we need to attach known fragments on both ends of this DNA fragment. cDNA that we have, right? We have to attach known fragments on both sides. If you have known fragments on both sides, you can design primers which are complementary to these, and then primers can come and attach. Then further steps can happen, whether it is amplification or it is sequencing. Both of them require adapter sequences. Okay. Now, one more concept is that it is very important that you have two different adapters attached on both sides of your DNA fragment. That means there could be a case where you have your DNA insert and adapter A and adapter A on both sides, or adapter B and adapter B on both sides. But the good fragments, the ones that are suitable for sequencing as well as amplification, is the one which has adapter A on one side and adapter B on the other side. Basically, two different types of adapters are required. Okay. Now, in case, I'm sure most of you might not have this doubt. In case you have a doubt that why should we require two different types of adapters, just to represent it very simply, I have drawn a diagram. Just wanted to explain what would happen if you join adapter A and adapter A. That means two same types of adapter on both ends. Let's have a look. Suppose this was your unknown cDNA fragment. Okay, this is your adapter A. It looks something like this: ATTCG and the complementary one. Now, once you attach this adapters on both sides, so one side will be 5-ATTCG over here. Here also from 5-ATTCG. The same thing is being attached over here and here. Now, what happens in the next step? In any PCR amplification, if you see, is the first step will be denaturation, right? You're going to denature the double-stranded DNA. Once you denature that, what could go wrong? If you observe these strands, right? This end and this end, ATTCG, CGAAT. Doesn't this look complementary to this particular area? What will happen? There's a high probability that your single strand, when your DNA molecule is denatured, these single strands can actually do self-priming. They can just go and, turn around and attach to themselves. What will happen if that happens? No amplification will take place, right? So that is one very simple way to explain why do we need this particular thing that, adapter A on one side and adapter B on another side. Okay, I hope that is clear. Adapters, they have a very special role to play in the whole sequencing process itself. Okay, so basically whatever you're going to attach, you're going to attach two different types of adapters. Whatever you're going to attach, they should have two basic elements. Okay, first is that it should be complementary to the adapters which are on a flow cell. After library preparation, we're going to do sequencing, right? Sequencing, if you look for a sequencing by synthesis method, it occurs in something called a flow cell. Okay, on the flow cell, there are a number of adapters which have been attached to it. Okay, covalently attached to the flow cell. Okay, so if your strand has to be sequenced, the strand has to go and attach itself on the flow cell to an adapter, which is complementary to it. So that is what is being told over here. It should be able to attach itself to the flow cell or bead. As we go further in the class, we'll understand exactly what I'm talking about. Don't worry. Second is primer annealing site for amplification and also for sequencing. So one is to attach. Another very important use of adapter is that it should have a sequence where a primer can come and attach itself. Okay. So that primer will be used in the process of amplification also, Also, when you go for sequencing also, that site will be used as a primer annealing site. And so that sequencing can also occur. Okay, so those two basic elements have to be there in your adapter. Okay, apart from this, you also have tags, barcodes, or indices. Okay, now why these are important? So most of the high throughput sequences, they produce millions of sequence reads in a single reaction or in a single run. In most of the cases, that the reads that we get are actually exceeding our need. We do not need so many reads for a particular sample. So in order to cut short the time as well as the cost of the experiment, what we can do is we can pool in different libraries from multiple experiments into a single sequencing reaction. This is called multiplexing. So to identify which experiment a given sequence comes from, each library is prepared using adapters which will contain a different tag. So these tags are called indices or barcodes. Okay, and these tags are typically 5 to 7 base pair sequences. And during the sequencing reaction, we not only read the sequence of the cDNA, along with it, we'll also read what is the sequence on these indices. So we can associate the insert as well as the tag or index together and find out from where a particular DNA insert or sample has come from. So that is the use of adding a barcode or index sequence along with our adapters. Okay. Also, with the help of these tags and barcodes now, or indices specifically, we can also carry out something called paired end sequencing. Okay, so we will be seeing what is paired end sequencing in the part 2 of this video. But please remember that adapter has a big role in actually carrying out the whole sequencing process. Okay. Now, while talking about adapters, we are going to specifically talk about Y adapters. That is commonly seen. You might have seen it in many diagrams, an adapter looking like this. Okay, so let us look at what is so special about these Y adapters. What you see over here is these two strands over here are diverging like this over here, right? It is just to denote that this area over here is complementary to each other, and these two particular strands are non-complementary. Okay, they're not complementary, so they are not forming a bond and forming the double-stranded structure. Up to here, it looks like a ladder, and then it diverges over here. That's basically because this portion is not complementary to this portion. So we design it in such a specific way that these two are not complementary to each other. Okay, let us look at how a typical Y adapter may look like. Okay, so suppose this is the left of the insert, this is the right of the insert. Insert is nothing but your cDNA. So left of the insert, right of the insert. That is where you're going to ligate these adapters, right? So you can see over here, if this is your left side of the insert, or this is your one adapter, right, left side adapter, you can see this particular portion over here. This is complementary, right? That is what is being denoted by this area. Okay, you can see G, C, T, A, they are complementary. Now just have a look at this area. They are completely non-complementary. C never binds with A, right? C needs a G. Same way A binds with T. So it is non-complementary. And that is why there will be no hydrogen bonds between these. Okay, basically, see, over here, if you have complementary bases, there are hydrogen bonds that are being formed, right? Between G and C, you have a triple bond, triple hydrogen bond. Between T and A, you have a double hydrogen bond, right? That will not be formed over here. And that is why it is represented like a Y. Okay, so this is how it will look. Same way for the right of the insert, if you look, this particular area is complementary, whereas you have a long area which is not complementary. And this particular area will also inculcate or have the indices in them. This is, you can see over here, index. Index is something which is specific to this particular adapter. Okay, these indices are generally 6 base pairs long, so you can have different combinations of ATCG in those 6 molecules, okay, or oligomers. Okay, now why these Y adapters are used? Okay, we'll be seeing that also. So remember, I told you that we have our DNA insert. Okay, I'll just show you the diagram. Yeah, we told that DNA insert will be there. You will be putting adapter A, adapter B, and we need fragments. The good fragments are those in which adapter A is ligated on one end and the second adapter B is on another end. So now what happens then is, usually what we do is we have DNA insert or cDNA fragments. We put in adapter A, we put in adapter B, and there's a lot of chances that you might not always get a good fragment. Okay, you might have fragments like this, where you have adapter A attached to both sides, or maybe adapter B attached to both sides. So these are useless to us. We need to select these particular adapters with two different adapters, right? Now, to overcome this particular problem of, leaving it to chance and then going and selecting the good fragments, we use something called Y adapters. The specialty of Y adapters is that once you attach it to your cDNA's ends and one round of amplification has been completed, when you look at the fragments, right, all the fragments will have two different types of adapters on each end. So you do not have to go for any kind of selection for finding out which is the good fragment. I will be showing you how that happens with a diagram. Now, I have drawn this diagram in such a way that the forward strand is shown in black color, the reverse strand is shown in brown color. I hope the color difference is evident on your screen. Why I have done this is so that you clearly understand where the forward strand is there, where the reverse strand is there, and actually how the adapters are being ligated. Okay, so I'll just go from top. RNA isolated, has been fragmented, has been reverse transcribed to cDNA. Now to these cDNAs, we are going to attach or ligate adapters. Okay, so you can see over here, you have your double-stranded DNA. And as I mentioned, this is forward, and suppose this is the reverse strand. Okay. First of all, you'll be polishing the ends. We talked about how end polishing is done. Then, along with that, you'll be adding an A, that is adenine on the 3' ends of these strands. So this is the 5' end, so this is 3' end, right? You add an adenine over here. Okay, you can see over here at the 3' ends. Now, to this, you'll be adding your Y adapter. This Y adapter has a T, that is thymine on this end. Okay, so what will happen? Thymine is complementary to your adenine, so it will easily come and join the DNA fragments, okay, with the help of AT bond. Okay, now please be aware that this is just one method by which adapters are ligated. There are numerous methods you will find to do adapter ligation, but this is the simplest one that I have selected, where you carry out 3' adenylation and then you add adapters which is having a DT overhang. Okay, you can see over here DT overhang, and that will form a complementary bond, and you have your double-stranded DNA and adapters attached. But adapter ligation does not end over here. You have to carry out one cycle of PCR. Okay, why? We'll see. So now, while doing the PCR, what happens? This is your double-stranded DNA with adapters over here. Why is this standing out like this? Because these are not complementary to each other. That's why I have put different colors over here. You can see blue and pink over here, right? This is not complementary. Okay. What will happen in the first step of PCR? Denaturation will occur. So your double-stranded DNA will be converted to single strand. So this is the first strand or the forward strand. I hope you can see this black and brown, right? This is why I just made the coloration like that. Okay. So what will be the next step after denaturation? You add the primers, right? Primers will come and attach to the 3' end of the strand. So this is the 5' end. So a primer will attach over here. Another primer will attach over here, which is complementary to this one. Okay. Third step is extension. So this particular strand will be extended. So since this is black, this will be the reverse, that is brown. Same way, this was your reverse strand, brown over here. The new strand has been synthesized, which will be black in color, which is complementary. I'm just trying to denote the complementarity. Now, when you look at these two copies, what do you notice? Look at this end, look at this end, these two ends. For this particular strand, look over here, look over here. If you look at these four different strands, each of them have a different adapter attached on both sides. That means adapter A here, adapter B here. Same way, A here, B here. For this, B here, A here. Again, B here, A here. Okay. So what happened? By the end of PCR, you actually have DNA molecules which are having two different adapters attached on each strand. Particularly on each strand we are looking, okay? Because one single strand is going to be taken and sequenced. You're not going to take both strands and sequence them together. When sequencing happens now, you do denaturation, you take only, or you work only on one of the strands at a time. Okay, so by the end of it, this is what we get. Okay, so that is the brilliant use of Y-adapters, we can say, in very simple terms. Okay, I hope that is clear. Okay, so that is a major step that has been covered. Adapters have been ligated to your cDNA. Okay, that is done. Now, before we jump into the amplification, I want you to know how or what exactly these adapters look like. Okay, let's just take one copy of DNA and then go further and analyze what and all components are there in these adapters. Okay, this is your forward strand, and suppose this is your reverse strand. This brown, black and brown, I have colored them likewise. Here, just to differentiate again, I have colored the area. Let us look at what it is. Now, when you go for sequencing, right, Illumina sequencing or something, you will hear about P5 adapters and P7 adapters. Basically, this is how that P5 or P7 adapters look like. I also want you to know this particular layout that I'm showing you is specifically keeping in mind that I'm going to do paired end sequencing for my mRNA. Okay, I have my mRNA, I got my cDNA, I will be doing paired end sequencing during the sequencing step. You can do single end sequencing, you can do paired end sequencing. But specifically, since I want to teach about paired end sequencing, I have shown an adapter which is compatible to paired end sequencing. So what and all components will be there in that. So if you understand paired end sequencing, you will easily know what is the difference in single end sequencing. Okay, there will be two types of adapters on the flow cell, P5 and complementary to P5 and P7. So your DNA insert will have on one end P5, on one end P7. These are basically to attach to the flow cell. Then they will have two different indices, I5 and I7. They will help in multiplexing. They will help in paired end sequencing to identify. Okay. Next, you will have the primer annealing site. And these sites are interchangeably used. Okay. Once it is used to read the sequence, once it is used to read the primer. For example, if your primer attaches over here, it will be read in this direction. Why will it be read in that direction? Because this is 3' right? Your new strand is always synthesized from 5' to 3'. So the complementary over here, it will read like this. So if you want to read the I7 index, primer will come and attach over here. If you want the read one, that is this DNA insert, if you want to sequence that, your primer is going to come and attach over here. Now, for conducting paired end sequencing, what you do is you invert the strand and make a complementary strand of it. Okay. So if you look at the complementary strand, this is your same P7, this is your same I7. But when you're going to take the read of this one, it will be from 5' to 3' in this direction, correct? So your primer for read to sequence will come and attach over here. And this will be read. Same way, if you want to read the I5 index, if you want to sequence the I5 index, the sequencing primer for I5 index will come and attach over here. And this particular index will be read. Okay. So what you will notice over here is a particular sequence itself. Here it is acting like the primer site for I7 index. Okay. Here it is acting as a primer for the read 2. Okay. The same thing, same over here. This particular place acts as a read 1 primer site. And this particular sequence also acts as the I5 index primer sequencing site. Okay. With that, I'll go to the next slide. Please do not be overwhelmed by this diagram. I assure you that in the part when I'm discussing paired end sequencing, all of this will make a lot of sense. So then you can just come back and listen to this part again. Definitely a lot of things will be clear for you. There it is. We have our library that is being prepared, right? Next, what you need to do is library cleanup and amplification has to be done. So earlier I had talked about this point, right? At every stage or every step of RNA-seq, you need to carry out quality check. You need to do cleanup because that is what will determine how well you get your results. Okay. So there is a process where you do library cleanup also. So in library cleanup, what you will try to do is you will find out whether if there are some cDNA fragments which have not been ligated with adapters, they will be discarded, or maybe we'll try to ligate adapters to them. Okay. And if there are primers which have not been, or primer sequences which have not been ligated properly, PCR is carried out to actually ligate those adapters properly. Okay. All those things are done during library cleanup. And the next step is you're going to do the amplification, because most of the times the initial starting sample might not be enough for us to carry out a sequencing reaction. You need multiple copies of the same RNA molecule that you have. From RNA, you get cDNA, so you need multiple copies of it. With one copy, you cannot carry out sequencing, right? You need multiple copies, and only then the intensity of the signal that we get during sequencing will be high enough to be noticeable, okay, to be captured. So for that, you need to carry out amplification. So with that, we are coming to the final step of library preparation. Okay, so the second step in RNA-seq is library preparation. In library preparation, the last step is amplification, you can say. What are the other things that we do? We do RNA fragmentation, we do reverse transcription, we do adapter ligation, and then we go on to amplification. Now, library amplification, as I told, it is required so that the received signal from the sequencer is strong enough to be detected accurately. Basically, it's for that. Okay. Now, different sequencing platforms carry out different types of PCR. You have emulsion PCR, solid sequencing, number 454 sequencing, ion torrent. They all make use of this PCR called emulsion PCR. So there, you do not have a flow cell. You have a bead. On that, your adapters are there, to which your cDNA fragments come and attach. Okay. But here, specifically for Illumina sequencing, since we are talking about that, we will be discussing about Bridge PCR. In Bridge PCR, basically there's a flow cell. Okay, so we'll be looking at that and we'll be studying what is Bridge PCR. This is also called cluster amplification in general terms. It is nothing but at one point you have multiple copies of a particular DNA fragments, or it forms a cluster. Okay. So this is how a flow cell typically looks like. Though nothing is seen to our eyes, like in each lane, there will be a lawn of oligonucleotides. These oligonucleotides are nothing but those adapter sequences. Okay. This over here, this flow cell is being represented by me like this. Okay. And on this flow cell, you will have, since we are using two different types of adapters, these adapters will be kind of, stuck. Oligonucleotides will be stuck covalently on these flow cells. You will have a lawn of oligonucleotides on the flow cell. Okay, so this is nothing but you had these P5 and P7 adapters on two sides, right? Those complementary sequences will be over here. Okay, P7 and P5. So that's why we are putting two different colors to denote that. So flow cell is coated with two types of adapters. So only that small portion will be there. Okay. Now, just to simplify things, I am representing our DNA fragment, cDNA fragment that is flanked by adapters in this way. Okay, one end is like this, one end is like this. You have two different adapters, and I have drawn this forward in blue color and your reverse strand in red color. Okay. Now, please remember, biadapter has a lot of components. It has primer sequences, it has barcodes, indices, and those sequences that attach to your flow cell. I have only shown that particular portion which will go and attach to the flow cell. Okay, just understand it is just a representation. So these are your adapters on both ends, and this particular strand has two different adapters. This particular strand has two different adapters. Okay. Now, as with PCR, what we discussed also earlier, what are the major three steps? Denaturation, annealing, and extension. That is what is going to take place here also. So the first step is denaturation, right? So if you have a double-stranded DNA, right, it will be denatured and you will have two single strands. Now, just to simplify and just to understand the concept, let us select one of these strands and then go forward. The same thing is repeated for the second strand also. Okay, so do not get confused. So let us just imagine that we are going to work only with this reverse strand, this red color strand that you see. You have double-stranded DNA, you have denatured it. We are going to select this reverse strand. Okay. Let us refer to these adapters for now as green and say purple. Okay. Just to differentiate it. Okay. So let us look at this reverse strand. It has at the 3' end the green adapter, at the 5' end the purple adapter. Okay. We are only going to focus on one strand. Denaturation has occurred. What these strands do is once it has been denatured, it will be made to flow over on the flow cell. Wherever the complementary nucleotides are there, they will go and attach themselves over there. For example, this is the green color adapter over here, oligonucleotide over here. please remember that this particular adapter is covalently attached to the flow cell. You can see over here, the attachment is covalent, okay? Whereas the new strand that has been denatured and which comes and attaches itself to the adapter over here is actually bound by hydrogen bonds, because this is complementary to this. There will be hydrogen bonds between these. So this is not kind of a permanent attachment. This is a very temporary attachment. So anytime we can denature it. Okay. So this one is always attached. The lawn, it has a covalently attached adapters, but the strand that is coming and attaching will be having hydrogen bonds. The same way the previous one, no? Forward strand will also go and find a purple adapter, right? Because it has to attach the 3' ends. Only then what will happen, if 3' end is attached to the primer, only then 5' to 3', the new strand can be synthesized. So same way, if you look at the forward strand, it will go and find out where the purple adapter is there. It will go and attach over there. Okay, but let us not focus on the forward strand right now. It's just to simplify the concept. Focus on one strand, the same thing happens with the next strand. Okay. So we have a reverse strand, which is attached over here. So after denaturation, single strand DNA comes and binds to the complementary adapters on the flow cell. Next, the second strand of DNA is synthesized, emerging from the adapter. So in the presence of DNA polymerase and DNTPs, what will happen? This strand over here kind of acts like the primer now, correct? So this is now acting like a primer. This particular adapter is acting like a primer. From here, the second strand of DNA is synthesized. So since this is red color, the complementary one, blue, is being synthesized over here. Just imagine this was your reverse strand, so a forward strand is being synthesized. That is why I have denoted two different colors, so that you always are aware what is happening. Okay. Once the strand has been completely synthesized, we carry out a procedure, denaturation procedure, and this original strand, right, this original reverse strand is washed off. The same will happen with the original forward strand also. After the second strand has been synthesized, that original forward strand is also washed off. So the point that you need to remember over here is the original cDNAs that you provide to the flow cells, those strands are all washed off. So whatever you get in the end after PCR amplification, those are all new strands. The original ones are not there. Okay. So this is washed off. And this is attached over here covalently, so it doesn't move anywhere. It is like firmly attached. Okay. Next, what will happen? This particular strand, right? 1 second. It has a 3' purple adapter over here. What will it do? It will go bend over and find out where is its complementary adapter on the flow cell. Wherever it finds the purple adapter, it will go and bend and form a bond over there. Okay, so that is what is being shown over here. And this is what is called the bridge formation, the beginning of bridge formation. And this is why this PCR technique is called bridge PCR. Okay, so you have this particular strand, it has bent over and it is, with hydrogen bonds, it has attached to this particular purple adapter. Also note, this purple adapter is covalently attached here, but this one is not attached. This is only attached with hydrogen bonds over here. Okay. Next, what will happen? So this is again the annealing step that has occurred, right? After annealing, what will happen? Extension will happen. So the second strand is synthesized. Now, since this is blue in color, complementary red is being synthesized. So from forward, you get the reverse strand that is being synthesized. Again, this is in the direction 5' to 3'. New strand is always synthesized from 5' to 3'. Okay. So bridge formation occurs. So newly formed strand bends and anneals to its complementary adapter. So new strand starts to form. What will happen when the strand is completely formed? You'll get something like this, right? You have a complete bridge formation. After this, what will happen? The bridge structure is dissociated. So all this takes place, under different chemical conditions, different thermal conditions. So we change the conditions in such a way that these strands act according to our needs. So once you change certain setups, what happens is these hydrogen bonds dissociate from each other. Okay, these hydrogen bonds over here, these hydrogen bonds over here, the ones between these all dissociate, and you get two different strands. These cannot move. Why can't they move? Because these particular adapters are actually covalently attached to the flow cell. You can't do anything. But these ends, since they were free, they just moved up and you have two strands. So you started with one strand. After end of one cycle, you're left with two different strands. Also note you have one forward strand and one reverse strand. But right now, both of them have the same direction, 5' to 3', 5' to 3'. Okay. The basis of PCR is repeating the cycles again and again and again. So now you have these two strands. These two strands will bend over, find out where their complementary adapters are on the flow cell, bend over, form or extend the second strand. The same process is repeated. So that is being shown over here. Purple will find out purple, green will find out green. They will be attached. The second strand will be synthesized. If it is forward, this will be reverse. If this is reverse, this will be forward strand. Finally, what will happen? This will all be extended. And you will have two bridges like this. And once they are dissociated, you will have 4 different strands. Over here, you can note 2 reverse strands are there, 2 forward strands are there. Okay, so this is just showing that again, if those 4 were going and attaching to their own respective complementary adapters, you will again have newer strands that have been synthesized after the bridge formation and then the dissociation. Okay, so bridge formation, new strand synthesis, and dissociation steps are repeated numerous times, forming a cluster. So this is how a kind of cluster looks like. At one point, if an original was added, once a copy has been made, that is washed off, and multiple copies of that particular strand are over here. Here at another point, another strand might have come and added. You might have cDNAs from different samples, right? So another cluster may be formed over here for a different strand, another one over here. Now, this is called multiplexing. Okay, on the same flow cell, you can do numerous experiments together. So that is where indices will help, because when you take it for sequencing, right, after sequencing has been done, you will be reading the indices or the indexes, index, on each one. And with that index, you'll be able to determine which particular sample it belongs to. So that is bridge amplification. Okay. So library preparation for Illumina protocols, it aims to generate cDNA fragments between 150 to 300 base pairs, which will be hybridized to a glass slide with microfluidic chambers or flow cell. Of those fragments, only the ends, that is 50 to 150 base pairs, will actually be sequenced. Okay, so the cDNA fragments that you have may be of 150 to 300 base pair sizes. Okay, but while you carry out the sequencing, you might only read, say, 50 to 150 from one end. Okay, so that is called the read length and everything, which we'll be discussing in the next class. So that was Bridge PCR and how clustered amplification is done. So we come to the end of part 2 of this session on RNA-seq. Today we learned about adapter ligation. We learned the importance of Y adapters. We learned the concept of bridge amplification. In case you found this video useful, please do let me know in the comment section. Also, please do subscribe to my channel. It will be a great help for me. Please do not forget to like this video too. I will see you soon with the part 3 of this video where we'll be discussing next generation sequencing. Until then, bye-bye.